This next section is going to focus on meiosis. So if you recall from mitosis, mitosis is when we took a normal cell with 46 chromosomes, we replicated the chromosome number to 92 chromosomes, and then we split that cell in half so that each cell, each daughter cell, had 46 chromosomes. We started with a diploid cell, and we ended with 2 diploid cells. In a process called mitosis, which is known as being diploid to diploid. When we go through meiosis, so that was mitosis. When we do meiosis, this is the production of haploid gametes. So for a person, we're talking about making sperm cells in a process called spermatogenesis, or making egg cells in a process called oogenesis. In meiosis, we're going to start the same way as mitosis. We start with a diploid cell with 46 chromosomes. Then we replicate it so that we have 92 chromosomes. Then we're going to split this so that we have 2 cells with 46 chromosomes. And then we go through another round of division so that we end up with 4 cells that have 23 chromosomes. So we're starting with diploid, and we're producing 4 haploid cells. Why can't we start with diploid and just simply split it in half so that we have 2 haploid cells? Well, as we go through here, you're going to learn about some of the things that have to happen, and those things would not be possible if we just simply took 1 cell and split it in half. The things that happen increase genetic diversity. It allows us to randomly reassort our chromosomes so that our offspring have unique traits from us. They have different combinations of traits from both our mom and our dad, so that we don't make identical replicas of ourselves when we reproduce. So not only are we increasing genetic variation through the process of combining a sperm and an egg cell, but the sheer process of creating a sperm and an egg cell creates for genetic diversity. Different combinations of genes end up in the gametes, and those traits get passed on to offspring. So, in a similar way, we go through the replication process. So we have replicated all of our chromosomes so that now we have chromosome 1 from dad, chromosome 1 from mom, and each one is replicated. So this is 1 copy of chromosome 1 from dad, this is 1 copy of chromosome 1 from dad. This is 1 copy of chromosome 1 from mom, this is 1 copy from chromosome 1 of mom. During the first phase, prophase 1 of meiosis, the chromosomes are going to condense and align align each other kind of in pairs so that this process called crossing over can happen. Crossing over is when the distal ends of adjacent chromosomes switch, so that you first start with complete chromosome from dad, complete chromosome from mom, and now these are going to flip over. Now think about this being your own cell. Now your cell has half of the information from your mom and has half of the information from your dad. So in your cell as an individual, this is one individual's genome. This came from that individual's dad. This came from that individual's mom. And now we're creating these hybrid chromosomes where it's all dad, mostly dad and part mom, mostly mom and part dad, and all mom. By doing this crossing over process, this increases the genetic variation of our offspring, because when we use this and we separate these out and use this sperm cell in order to produce offspring, this grandchild, so your child, this is your dad, that would be, you are a combination of all of that, and then you would produce a sperm or an egg from that. The grandchild from this would be almost 100% like grandpa, or at least 50% of that particular chromosome. Now, if this one was used to produce your offspring, the grandchild from this, or your child from this, would be a lot like your dad, but also part of your mom, just for this chromosome location. If this one was used, again, it would be mostly from your mom, and then part from your dad, and this would be a majority from your mom. So these particular traits for this specific chromosome location would be inherited that way. So crossing over is a mix of the distal ends of adjacent chromosomes to produce these hybrid chromosomes to increase genetic variation. Now, while doing this, each chromosome independently assorts, and that's kind of the law of independent assortment. There is no tie saying that an individual will inherit this form of the chromosome for every single chromosome locus. So this is chromosome 1, this is chromosome 1, this is chromosome 1, and this is chromosome 1. The baby can get any number of that chromosome 1. For chromosome 2, they can get any variation of chromosome 2. And chromosome 3, they can get any variation, and so forth. Because each chromosome is inherited independently, and they're not tied to each other, we have the law of independent assortment. So during prophase 1, crossing over happens, the chromosomes condense, and that nuclear envelope disappears, just like it did in prophase of mitosis. During metaphase 1, we have our cell. The chromosomes are going to align down the center of the cell, just like they did in prophase, in mitosis, except this time they're going to line up by pair. So we have our crossed over chromosomes. They line up in pairs like this, where chromosome 1s are in pairs, chromosome 2 in pairs, 3, 4, all the way through the 23 sets of chromosomes. And the spindle is going to attach, so the spindle complex is going to attach to the chromosomes. But they align down the center of the cell. During anaphase 1, the chromosomes are going to split and migrate toward opposite ends of the cell. But you'll notice that they don't all split down the middle. like they did in mitosis. If we look at the picture of what happened in mitosis, all of the chromosomes align straight down the center, and during anaphase, they split, where half went to one side, half went to the other. Now going over to meiosis, they still aligned in the center, but they aligned in pairs. And these are all called tetrads, and tetra is for 4, so they're tetrads of chromosomes that align down the center. And then when they split, these pairs of chromosomes are going to go toward opposite poles. Telophase 1, new nuclear envelopes form. And remember, these are crossed over chromosomes. And then cytokinesis 1, you're going to end with 2 cells that have 46 chromosomes. And I'm only drawing 2 pairs of chromosomes, but drawing 46 would take forever. After we go through cytokinesis 1, we immediately jump into prophase 1. And in prophase 1, those nuclear envelopes are going to immediately go away. So why form them if they're just going to go away in the very next step? Because when an egg is produced, it doesn't complete meiosis until the egg is actually fertilized. So it allows for more control of the process of spermatogenesis and oogenesis. So we have our chromosomes that condense. Now, crossing over only happens in prophase 1, so we're not going to have a second crossing over event. During metaphase 2, we have them align in the center of the cell again. And the spindle fiber will form. During anaphase 2, they'll migrate toward opposite ends of the cell. And then during telophase 2, new nuclear envelopes are going to form. And in cytokinesis 2, you're going to end with 4 haploid cells. And due to the fact that these went through crossing over, you're going to see some differences in the haploid cells. If these are your sperm cells, or if these are your egg cells, this contains mostly information from your dad. Which means if you use this gamete in order to produce your offspring, your baby is going to look a majority like your dad, or at least 50% of your dad. If you use this gamete, your baby is going to be a combination of both your mom and dad. If you use this gamete, your baby is going to be a combination of both your mom and dad. And if you use this gamete, your baby is going to look a majority like your mom, or at least 50% like your mom. Now this gamete is going to be combined with the gamete from the opposite sex, so they're going to be 50% from that side of the family and 50% from this side of the family. But this rarely happens where one egg is produced and contains 100% all of the information from your dad. It's a random combination. So a more realistic picture would be 46 chromosomes with different amounts of material from both your mom and your dad. And because of that, and because of that law of independent assortment, we have all sorts of different combinations of our offspring that can be produced.